Thank you all so much for taking the time to listen to my presentation today. I know it's a little bit later for you there, and I appreciate you hanging around. I also wanted to mention I do have a screen here where I can see all of your faces. So if you do feel like sharing your video, I really appreciate it because I do take cues from my audience when I'm speaking. And I can see you right over here. Thank you. So I think recently in biological sciences have made a lot of significant advancements in integrating data across these different levels of biological organization. And a lot of this integration, the goal is to improve predictability and elucidate these rules of life. And we all come with different perspectives towards understanding biological processes. Some of us come from an ecosystem community perspective. Some of us come from a population organism perspective, and some from a molecular perspective, and some from a phylogenetic perspective. But regardless of our perspective, with this explosion of data, we're all typically developing or applying increasingly complex models. So here's what I say is a typical approach that's used in our fields. There's some biological process we want to understand. We collect data to try to understand that process. We apply statistical models that are becoming increasingly complex to analyze this data. And then we build a picture or prediction of how that biological process works. And one thing I'm really interested in is inference. How accurate is our understanding of that process? When does statistics mislead us? And what are we missing? And I'm interested in these ideas about inference in the context of global change. How does evolutionary history shape the genetic architecture of adaptation? And then how do these evolved architectures in turn shape the short and long-term responses to evolutionary change? Can we use this genetic information to predict how populations will be more vulnerable to certain kinds of climate change? And if we could make these predictions, it would allow us to manage populations better under global change. But to return these ideas about inference, in many cases, answering these questions first requires understanding if the methods we use to do inference are misleading us. So I have empirical expertise in a number of different systems, mostly in marine invertebrates and fish, but I've also collaborated with tree biologists. A lot of the species that I work on are long-lived. They have large population size, they have high dispersal potential, and they're horrible experimental systems, to be honest. But I love working in marine systems, and a common theme in my empirical research across these systems is the application of principles in population genetics and genomics. So to better understand the influence of climate change on species and to explore ideas about inference and how accurate or inaccurate it may be, my lab integrates principles in statistical genomics and data science with evolutionary theory and simulation and molecular ecology and experiments. So today, my talk is divided into 3 sections. In the first part, I'm going to highlight an area where I think we're overlooking in terms of studying plasticity and responses to climate change. And this is in the study of gradient variation, including countergradient and cogradient, the evolution of these patterns. In the second part, I'm going to highlight how testing methods with simulations is a powerful way to validate or invalidate different models, and share some insights with you in trying to understand the statistical analysis of inversions in population genetics and this new concept of genomic vulnerability that's becoming really popular in the literature. So let's get started talking about plasticity. So this first part of my talk is going to highlight our research into the evolution of covariance between genotypic and environmental effects on phenotypes. And this is known as gradient variation. And this work has been part of a synthesis project under the Research Coordinated Network for Evolution in Changing Seas. So the National Science Foundation here in the US funds these networks to bring together different kinds, people in different fields. And this network's trying to bring together marine scientists with evolutionary biologists. And it's an open network, so if you guys are interested, you're more than welcome to join. And we love having people who don't work in marine systems, because the whole idea is to integrate principles across different fields. So geographic variation in Clines has long served as an observational basis for ecological and evolutionary studies. And to determine the genetic and environmental drivers of these Clines, we either use reciprocal transplant designs or common garden experiments. And we measure some phenotypes or some genetic patterns. And from there, we try to understand if the environment only, the genotype only, or some combination of genotype and environment are driving the observations that we see in nature. And these genotype by environment interactions are really important drivers of phenotypic variation, because they indicate that the influence of genetics on the phenotype is varying across environments. So we need both information about the genotype and the environment to make any kind of prediction about what the phenotype is going to be. But these G by E interactions are not the only way that genotype and environment can be associated to affect phenotype. There also can be covariance that evolves between genotypic and environmental effects on phenotypes. This covariance was first observed by Levins in 1968 in Drosophila melanogaster. It was then observed by Bervin in 1973 in frogs, and probably most famously by Conover and Present in 1990 in these Atlantic silver sides, which are these estuarine fish that are on the east coast of the States. And then recently, my postdoc also observed these patterns of covariance in frogs that she was studying between that had evolved to saltwater environments in salt marshes along the east coast here. So we've known about this covariance for over 50 years. We have examples of it in nature. It may be as important as genetic by environment interactions. So why aren't we all measuring it? Well, how did researchers identify this covariance? And the problem was there was really no way to measure it. The way people have been talking about this subject and thinking about it has been to use these visual patterns in reaction norms. So, for example, if they saw a pattern like this, where we have a genotype that was collected from one environment and a genotype that was collected from a second environment, we see that the environmental influence on the phenotype is positive. So the slope of the reaction norm here is positive. And also we see that the genotypic effect on the phenotype is also positive. So as we move from the genotype in environment 1 to the genotype in environment 2, we see an increase in the phenotypic variation. And we call this co-gradient variation because the genotypic and environmental influences are in the same direction. And this usually lends itself to observing strong Clines on the landscape, and when you look at the genotypes in their home populations. In contrast, counter-gradient variation, you might notice now the blue line is on the bottom and the green line is on the top. So we have negative influence of genotype on environment. And this is when those two effects oppose each other. It's counter-gradient variation. And in this case, when we go and survey genotypes on the landscape, we may not see a phenotypic line across that environmental gradient. And this was pointed out by Conover and Schultz in 1995. We may be missing a lot of these examples because we don't observe phenotypic differences across an environmental cline. So on the left here, we have positive covariance between genotype and environment, and on the right, we have negative covariance between genotype and environment. But without a quantitative method, this approach, this kind of visual approach has limits. What if we do a reciprocal transplant with 4 genotypes and 4 environments? Is this co-gradient or counter-gradient variation? And what if there's some genetic by-environment interactions as well? And we can't, without a method, we can't test if these visual patterns are real. We can't answer bigger questions about covariance, such as, what are the implications of this in populations and how they evolve and how they respond to climate change. So Falconer in 1989, in the famous Introduction to Quantitative Genetics textbook, pointed out that phenotypic variance can be broken into these different component parts: environmental variance, genetic variance, genetic by environment interactions, and this COV-GE, this covariance that I've been talking about. But he didn't describe how to estimate this COV-GE in the presence of residual variation and genetic by environment interactions. And so to our knowledge, nobody has actually estimated this variance component. So we were thinking, well, if we can't estimate the variance component, maybe we can just estimate the effect size of the COV-GE from data. And so to give you an intuition of the sampling estimate that we developed, I'm going to walk you through how we estimate the environmental and genetic effects on the phenotype. So we have some data that we collect in a common garden or reciprocal transplant experiment. We ask first, what are the genotypic effects on the phenotype? So we get the effect of the genotype, G1 and G2. Then we ask, what are the environmental effects on the phenotype? So the environmental effect from environment 1 is the average of the 2 genotypes, and then environment 2. And now we have a measure of covariance between these genetic and environmental effects. And this is the sampling estimate that we developed. And it looks kind of like a covariance, but there are some important differences here. The first one is this I. This is an indicator variable because we only want to count terms towards the covariance when those genotypic and environmental influences match. So if G1 is with E1, it counts. If G2 is with E2, it counts towards that calculation. The other interesting thing is that we have a unique kind of standardization here. And the reason is because these genotypic and environmental influences on phenotypes are non-independent. We have to use the maximum of the variance in the group means of either the genotypes or the environments, whichever one is greater, to do the standardization. And this essentially preserves that relationship between genotype and environment because they are non-independent. So this metric gives us a value between -1 and +1, just like a correlation that can indicate the degree of covariance or COV-GE in the data. So we use bootstrapping to develop 95% or to calculate 95% confidence intervals. And we use permutation for hypothesis testing. So the null hypothesis here is that COV-GE, or this covariance between genetic and environment, is 0. So to validate this statistic, we simulated 30,000 plus patterns that might arise from different experimental designs used in common garden and reciprocal transplant experiments. So here's an example of reciprocal transplant with 4 different environments. And this data gives a positive covariance. We also get confidence intervals, and we get a p-value for that that COVE-GE. We also simulated data with genetic bioenvironment interactions and covariance, so there can be both present in a data set. So this is another example of covariance, which by eye might not be as evident, but is a very strong covariance pattern. We also simulated these paired common garden experiments. So maybe you have, like Molly did, you have multiple replicates of the evolutionary process. In her case, she had adaptation of freshwater frogs to saltwater marshes on the East Coast. And you raise them in freshwater and saltwater environments, or whatever the two environments are in your data. And this is another kind of design that we simulated that you can calculate the statistic from. So now we're in a position to ask, what kinds of experimental designs do we need to do to really study and get a good handle of this COVE-GE in nature? So we did, using all those simulations, we did a power analysis, and the results are plotted in this heat map. So let's say you go out and you sample 2 genotypes, and you sample 2 individuals per genotype, or you sample 8, well, that was too few, so we didn't even do that, but we sample 8 individuals per genotype, or 16, or you go out and you sample 8 genotypes from 8 different locations, and you sample 4 individuals from each one. You know, what is your power to detect COVE-GE in a full reciprocal transplant design? So in this case, very large sample sizes are typically needed to get power. I mean, it depends a lot on like how much standard error there is in the data, etc. But typically large sample sizes are needed. And this was also true for the paired common garden design. And another thing to point out is that, you know, these most common designs are typically these 2x2 reciprocal transplants, where you have like maybe a high altitude and a low altitude genotype, and you raise them at high and low altitudes. These would have very low power to detect this COVE-GE. So in addition, we've been applying the metric to empirical data. So here is an example from Molly's frogs that I was telling you about, where they're adapting to saltwater coastal marshes. And applied up to her data, we get a negative COVE-GE, so this is counter gradient variation, and this is a significant pattern. We've also applied it to our other collaborator, Jeff Trussel's data, and in this data set, he also has a pretty strong pattern of counter gradient variation, so that's this negative covariance, but it's not significant. So again, this is a kind of design that has low power because it's only a 2 genotype by 2 environment reciprocal transplant experiment. So now we're in a position to do a meta-analysis and see if this COVE-GE is as common as genetic bioenvironment interactions in the literature. And this is interesting because, you know, a lot of people aren't interpreting their data in this context. So I didn't get a chance to talk about this today, but we also developed a standardized metric of genetic bioenvironment interactions for this meta-analysis. Just think about, like, what do you think this pattern is going to look like? So we started with 4,000 initial studies based on like looking for search terms like local adaptation and reciprocal transplant experiments. It turned out not too many of those studies, about 60 or 80 of them had the kind of data that would meet the criteria for applying this statistic, but most of those also measured multiple traits on their study species. So in some, we have a few hundred total traits in this meta-analysis. All right, and so here's the pattern. The initial pattern is preliminary, but it's all over the place. So we see cases, we see some systems where there's high covariance and high genetic bioenvironment interactions, some where there's covariance and no genetic bioenvironment interactions in that trait. So it's unclear yet how these patterns are going to hold up when we test them statistically. But it seems that from this analysis that COVE-GE is at least as common as genetic bioenvironment interactions in the species that we study. So in terms of plasticity and climate change, I think this evolution of covariance between genetic and environment interactions are something that we're missing. But as I mentioned, we only simulated patterns to apply our statistics to. We still don't have a good understanding of how these patterns actually evolve. And then how does it change predictions for how a population might respond to climate change? Whether a trait has a countergradient or cogradient pattern could have different implications for how that species evolves in response to climate change. So these are still open questions that my lab's starting to work on. So now I'm going to move into the second part of my talk and transition more into talking about model validation in population genetics. And my research has been really interested in pitfalls that can happen when genome scanning. So genome scans are these statistical tests that we use to try to understand the genetic basis of a trait or the association between some genotype and the environment, or some genetic differences between populations. So in general, we're scanning along a genome and we're looking for these areas in the genome that are outliers. And so they're doing something that's different from the rest of the genome, and often we're trying to test if that is under selection. And earlier on in my career, I had looked at the effect of non-independence in the data in terms of complex demographies. So, genome scans, some of the earlier methods assume these island models where populations were essentially equally dependent on each other. But in reality, a lot of the species that we study have more complex demographies. Some populations that we sample are more closely related evolutionarily than other populations. And it's this complexity that can confound some genome scan methods. So for example here, I'm not going to go into the details, but all of the methods that we've tested have low false positive rates when we simulate island model data. But as we simulate more and more complex demographies, some of these methods, especially the ones that are assuming an island model, have much higher false positive rates. So for this reason, I advocate for model validation, and this is central to all the research that's been being done in my lab. So the basic idea is that we simulate some data so we know the truth in the data that we're analyzing. We do the analysis in the same way we might do the analysis on the empirical data, and then we evaluate, given that truth known in the simulations, how do the different methods perform. But there's a catch here, which is the simulations have to capture some realism in the data, and they don't follow the statistical model that you're testing. And I think if the field is guilty of anything, sometimes it's guilty of just testing methods on simulations that perfectly meet the assumptions of that method. And just an interesting backstory, I became interested in model validation when I was working on my PhD, and I was studying fish recruitment. And we were testing if some of these large recruitment pulses of fish that we were finding were siblings. And I was putting my data into this program, and somebody really smart said to me, well, you should randomize your data and put it into that program and see if it tells you they're siblings. So I did that, and I was like, well, that's a waste of time. Of course, that's not going to tell me that it's siblings, it's random data. So I put my random data in, and sure enough, that person was right. The program output some siblings. And the program didn't output as many sibling pairs as we observed in the data. So I knew some of the siblings I observed in my data were real, but it made me realize, that exercise made me realize that I couldn't tell which ones were real and which ones were randomly generated by the program. So that's just a little background of how I kind of started to get interested in this idea about inference and methods. So today I'm going to talk about two insights that my lab's getting from these kinds of simulation studies. And the first is with respect to inversions. So inversions have been implicated in adaptation in a number of different species, and from trout to ants to stick insects to butterflies to birds and vertebrates. I mean, it's just amazing how often inversions are implicated in adaptation. And an inversion is a place in the genome where the DNA has essentially flipped. And it doesn't affect the function of those genes because all of those genes are still present in the genome. But the way that recombination works, it's looking for homologous sequences. And so when it gets to an inverted region, the recombination machinery is like, well, this doesn't match, so we're not going to have a break. We're not going to have a recombination event here. So these inverted regions have much lower recombination rates compared to the rest of the genome. And so the whole idea behind why inversions are implicated in adaptation is because this lower recombination reduces gene flow between these inversion haplotypes. And it allows one haplotype to adapt to one kind of environment and the other haplotype to adapt to a different kind of environment. But a lot of these methods, a lot of these studies are using these genome scans to implicate inversions and adaptation. So here's just an example from Atlantic cod where they see there's this higher FST in this region that is an inversion, and also this higher linkage disequilibrium in this region that's an inversion in the genome. But there's an issue, and that's because this recombination variation is another source of non-independence in the data, and it's not clear if we're accounting for properly when we're doing these genome scans. So just like when comparing an island model to a complex tomography, if we analyze data, unlinked loci in our data, which are, they're not totally independent because they're all in the same genome, but they're quasi-independent, they're mostly independent, versus, just putting everything into a program, all the data that we collect from a genome. Some of those loci are going to be closely linked and have less recombination between them and be more dependent to each other than others. So it's a kind of an analogous thing to thinking about populations. So I started just with the simple question is, what is the effect of neutral recombination variation on genome scans? So I created these simulations in continuous space. So there's a couple of mountaintops in this data, and there's these little populations of individuals that form patches in my simulation. And it's a quantitative genetic model. So it's a whole genome model. There's effects, there's these quantitative trait nucleotides that have additive effects on a phenotype. And then this phenotype is under selection with different optimums according to the colors in this figure on the left. And I simulated a genome with 10 linkage groups. And each linkage group had something a little bit different going on. So linkage group 1 was neutral. Linkage group 10 was also neutral. Linkage groups 2 and 3 were where these quantitative trait nucleotides could evolve to have effects on the trait that adapted to the environment. Linkage group 4 had a selective sweep. 5 and 6 had these regions of background selection. Linkage group 7 had a neutral inversion. Linkage group 8 had a centromere, which was just a region of low recombination. And then I had a variable recombination linkage group. But all of the recombination variation was neutral. So I expected when I put data into this, so I was just to say what evolved in these populations was just isolation by distance. So I expected, some kind of isolation by distance population structure when looking at a principal components analysis. But when I took the data from these simulations, I just plopped them into the like a genome scan method or principal components analysis. This was the observed population structure that I got. And what was happening was this non-independence between loci within inversions was driving the principal component. So here you can see that the inversion genotype was separating out the population structure. And this was happening despite the fact that those loci were less than 10% of the data that I was putting into the principal components analysis. In turn, this then had an effect on genome scan methods that use principal components to control for population structure. So if we look at one of these outlier detection methods that's based on principal components, this one's called PC-Adapt, you would see that it would give you these outliers in the neutral inversion and in the region of low recombination. And that's because of the way the method was biased by these this non-independence between loci. So that's a naive practice, just plop your data in. But there's a better practice or a better principle, which is you can thin the SNPs or the single nucleotide polymorphisms for linkage disequilibrium. Then you conduct this neutral parameterization. So you estimate the nomography on these thin set of quasi-independent SNPs, and then conduct the genome scan on all the SNPs using this parameterized demography. So on the top row here is the results that I was showing you, and the population structure estimated from principal components with this naive approach. And then on the bottom is this best practice. So when you thin the SNPs to estimate structure and then use that to conduct the test on all of the SNPs, you can recover the correct demography, and then you get outliers at these loci that have effects on the phenotype that's adapting to the environment. So here the orange arrows indicate all of the quantitative trait nucleotides, and the thicker the arrow, the larger the effect that evolved of that nucleotide on that trait. So you get signals in the right places with the best practice. So what is the effect of neutral recombination on genome scans? Well, none if the best practices are used. And so, this was just neutral recombination variation. So then the next question is, well, can we reliably detect adaptive inversions? Well, to answer this question, we have to simulate genomes with realistic patterns of inversions. And when we look in the literature, we see that a lot of these studies that find inversions have genomes with multiple inversions implicated in adaptation. Those inversions are really large and they're really old. And so, we also need to answer this question of like, why do we observe this? How can we generate simulations that do this? So on the top here is an example in Atlantic cod, where you can see there's 4 inversions that are FST outliers between migratory and non-migratory ecotypes. On the bottom here are FST outliers between snails that have snail ecotypes that have adapted to wave exposed and calm shorelines. And again, you see these really high peaks of FST and multiple inversions implicated in adaptation in the genome. So we took this quantitative genetic model, we simplified it to a 2-patch model, and this is a study led by my graduate student, Sarah Schall, in collaboration with Ben Haller, who's the developer of SLIM. So in this model, I'm just going to play this video. Oh, go back. There it goes. We have 21 chromosomes in our genomes, and it's called inversion invasions because we just let inversions mutate and invade the genome. So this is showing every 200 generations, all of the inversions that arise on the genetic map across all of the individuals in our simulation. So there's a lot going on in this is presenting a lot more information. These aren't all arising in the same individual, right? These are popping in and out in different individuals. And there's a 10,000 generation burn-in, after which there is this selection by the environment. So one patch has the optimum phenotype at +1, the other one has an optimum phenotype at -1. And over time in this simulation, we're seeing multiple inversions becoming segregating in the population and differentiating between those the genomes that are adapted to those two patches. So now we can ask with these simulations, like, what conditions do inversions actually help and aid in local adaptation? So on the x-axis here is generation time. On the y-axis is the degree of local adaptation. So a local adaptation of 1 is like perfect fitness in its home patch and like horrible fitness in the other patch. So this is a subset of our simulation. So we have no inversions on the left, medium inversion mutation rate in the middle, and then a higher inversion mutation rate on the right. And the different colored lines are different migration rates. So just to orient you here, at low migration rates, we don't see any differences between these three panels. So inversions aren't really helping with local adaptation under these conditions. At intermediate migration rates, however, this intermediate blue line here, we see that inversions are having a huge effect in how much the population adapts to the environment. And at these lower migration rates, or sorry, even the highest, very high migration rates, so 0.4, it's almost panmixia, at the highest inversion mutation rate, we see that inversions can allow adaptation to occur when otherwise it would not. And I will say that these are a subset of our simulations that have highly polygenic architectures. So inversions are helping in adaptation when there's a lot of loci with really small effects on the phenotype, and the phenotypes are under strong selection in each patch. So this scenario right here is really interesting to me because there's such a huge effect of inversion mutation rate on the amount of local adaptation that is achieved. So we're going to dig a little bit deeper into this scenario here. So this is just another video, and this is the whole genome. So all 21 chromosomes are just back to back here. On the y-axis is the number of QTNs within that inversion, and then on the x-axis, or on the top graph, is FST. And so over that first 10,000 generations is the burn-in, and you can see all these little hatch marks down here are different inversions that arise, and the color of green indicates their frequency. So if they're at a frequency of 0.5, they're a little bit greener. And about at this point, after 6,000 generations of adaptation to the environment, something really interesting happens, which is several inversions arise and differentiate between those two patches at the same time. And we've dug into this a little bit more, and it's a combination of when those inversions first mutate, they capture some loci that puts the population kind of like in the right direction for that patch. And then those QTNs, as they mutate, will accumulate in that inversion through time until the population reaches an equilibrium. And we have control simulation. So here's just an example of the FST in these different inversions. The red ones are inside the inversions. The blue dots are outside of the inversions. And in this simulation, we had 6 inversions arise and be the genetic basis of adaptation in this simulation. And just to control for potential effects of genetic drifts, we have simulations with no selection just to see if genetic drift could cause these patterns, and it doesn't. In addition, if we look at adaptive versus non-adaptive inversions and we compare them to our control simulations without selection, we see that the adaptive inversions have they're much older than the non-adaptive inversions. They're they tend to be larger than the non-adaptive and the no selection inversions. And they also produce a realistic haplotype structure. So to look at the haplotype structure, we created these heat maps. And this is just a heat map of several sections of the genome. And you can see the inversions here as these kind of blocks of LD. And we have population 1 individuals in the top and population 2 individuals in the bottom. The homozygote reference allele is dark red, heterozygotes are in yellow, and the homozygous alternate allele is in lighter yellow. And I don't have time to talk about this today, but I've just compared this kind of like haplotype structure to something that we find in the eastern oyster. So I've been working on the eastern oyster as part of the breeding consortium. We're trying to understand how oysters are adapted to the environment and disease. And this also has like an isolation by distance population structure. And we found a few inversions in the genome that have really high FST values, which are these different colors here at the top. And here's the haplotype structure that we observe in the real data. So this is the simulated data. This is the real data. I know the color scheme is a little bit different here, but I'm pretty happy with this, you know, like I'm like, okay, we're creating simulations that are capturing like some aspects of what we see in real data. So I was pretty happy when I saw this. So now we can ask, well, are these inversions like confounding outlier tests? Do outlier tests working in detecting these? And when we apply that best practice that I mentioned before, which is you thin the SNPs for linkage disequilibrium first, estimate the neutral demography, and then use that neutral parameterization to conduct that genome scan and all of the data, you can recover signals in the correct places in the genome where those inversions are underlying adaptation. So, you know, we're getting closer to publishing this. We're still looking across this whole parameter space to see if these conclusions hold. But, you know, so far we can say that we observe these genomes with multiple large and old inversions when there's moderate to high gene flow, high mutation rates, and highly polygenic architecture. So this kind of lends support to that supergene hypothesis. It is often cited with inversions. And can we reliably detect adaptive inversions? So far, everything we've looked at indicates yes, if we use this best practice that I just described. So now I'm nearing the end of my talk. I just have a short section at the end to highlight sort of this growing interest in estimating genomic vulnerability. So this idea of genomic vulnerability was presented in this 2018 science paper. And what they did was take landscape genetic data and put it into a machine learning algorithm that output what they called a genomic vulnerability. And they found that when they looked at the population trend of these birds relative to a genomic vulnerability, which is this idea of like the mismatch between the genetics of the population and the environment that has changed, that they found this negative relationship. And so they use this as evidence that there's some relationship, that this genomic vulnerability can be used to identify populations that are maladapted to the climate. And then another study by many of the same authors came out the same year in Ecology Letters, where they also found this negative relationship between population size and genomic vulnerability. And the method they were using to estimate this genomic vulnerability is called gradient forest. And this idea was first presented by Fitzpatrick and Keller 3 years before those studies were published in this Ecology Letters paper. And what this method does is output this cumulative importance as a function of the environment for each locus in the genome. So each line here is a locus in the genome. And loci that are under selection to the environment have these steeper and higher cumulative importance functions. And so the idea of this genomic vulnerability was first called a genetic offset by Fitzpatrick and Keller. It was rebranded a genomic vulnerability by Bay et al. And Ruegg et al. And it's based on this cumulative importance function. So let's say, for example, this is a cumulative importance function. Once you get a summary across all of the loci in your genome, it's driven, in their minds, it's driven by loci that underlie adaptation to the environment. And therefore, the distance in this cumulative importance between a population at a current temperature and a population A prime at a future temperature, this vertical distance is the amount of, this is what they're saying, is the amount of genetic change that would be necessary in that population to respond, or for that population to be adapted to that future environment. Does that make sense? Right, some people with their videos on nodded, so I'll keep going. For the purposes of my talk, I'm just going to call this a gradient forest offset because that's all it is. It comes specifically from this method. So I call it a GF offset. So we're trying to validate this with simulations, and this is a project led by my former postdoc, Elke Lorison, in addition with Mats Fitzpatrick and Steve Keller, who were the original authors of that Fitzpatrick and Keller paper that I mentioned. So the first thing to understand about this cumulative importance is that it's modeling the turnover in allele frequencies on an environmental gradient. So let's take an allele that starts at a high frequency at negative values of the environment and ends at a low frequency at positive values in the environment. That has a cumulative importance function that looks like this. We can take an allele that has the complete opposite direction, maybe even opposite effects on fitness, and it outputs the same exact cumulative importance function. So right there is a sign that, like, we shouldn't trust this method to tell us anything about fitness. Here's another example of alleles that decrease and increase in frequency, or increase and decrease in frequency. Here, the genetic offset is not changing very much at these intermediate values of environment because there's not a lot of allele frequency change at intermediate values of the environment. Another thing to notice is that the allele frequency is exactly the same at very negative and very positive values of the environment, but there's a huge offset here. So again, it's modeling turnover in allele frequencies. So my question when I read these papers was, well, could these patterns arise because of genetic drift and not because of signals of selection? So to answer this question, we created simulations from low population size, high population size along an environmental gradient, constant population size, and then high to low. And these were completely neutral simulations. We had no selection in these simulations at all. So here are the cumulative importance functions that we get out of these neutral simulations. So here we have the equal population sizes in the middle. So more or less a linear relationship between cumulative importance in the environment. With decreasing population size, we see a curve that looks like this. All right, so are you guys ready? I'm going to walk you through it. So what happens here is high population size. We have low genetic drift. We have low allele frequency turnover. So we get lower genetic offset. At the other end of the landscape, we have smaller population size. We have higher genetic drift. We have higher allele frequency turnover. So we get a steeper genetic offset. All right, we're going to do it again. So we're going to do it for increasing n. So at this end of the landscape now, we have smaller population size, higher genetic drift, higher allele frequency turnover, higher genetic offset. At this end of the landscape now, on the right side, we have higher population size, we have lower genetic drift, we have less allele frequency turnover, and we have lower genetic offset. Every time I do that, it's exhausting. Okay, so now we can look at, okay, what is the relationship between genetic offset and population size. And we can do this for the increasing N and decreasing N scenarios. In both scenarios, we see there's a negative relationship between the population size and the genetic offset. Negative. So could these patterns between genetic offset and population size arise because of genetic drift and not selection? Yes, empirical studies find exactly what we expect based on neutrality. So this raises some questions about, what does this measure of, quote, genomic vulnerability mean? What is it telling us? And can we actually use it to estimate something about how adaptive a population is to climate change? So I have a lot more to say about this subject. I don't have time to talk about today's. It's a very nuanced yes, and I'm happy to answer more questions if you guys want to chat. But what's interesting now is that there's this rise of genetic offset methods in the literature. And I'm not going to go into all of these methods, just that, people are trying to use genomics to predict something about how a population will be adapted to climate change. And so far, these are not being tested on simulations. And the couple that have, I would call them, they're being tested on friendly simulations. They're not really being challenged. So I was just going to mention, this is an area that, my lab's interested in working on more. So in conclusion, I think that, with regards to plasticity and climate change, gradient variation is something that we're missing in this, but now we have a method that we can use to estimate covariance between genotypic and environment influences on phenotypes. Studying inversions is really cool, and with best practices, we can make more accurate influence about their role in adaptation. And then finally, we should apply caution when using these genetic offset methods until we understand them better. Especially because, patterns between population size and, this genomic vulnerability can arise from neutrality. So in conclusion, I started this seminar by highlighting the different perspectives that we all take. And I think it's interesting to think about inference in this time of big data. And I just want to make a little plug that I think that model validation is a really important aspect of validating these more and more complex models that we're using in population genetics. So with that, I've definitely talked long enough. Thank you all so much for listening and having me here today. And I'm excited to meet and talk with many of you over the next day or so. And I'd be happy to take your questions. Thank you.